In this video, we're going to continue our discussion of controlling microbial growth. In particular, we're going to be looking at the antimicrobial drugs. Now, before we get into the antibiotics and the antiviral drugs, let's talk about the goal of why we use antimicrobial drug therapy. We're using it because drugs can target specific interactions with the bacteria, viruses, etc. So the goal is that we're using these drugs to target, to destroy the infectious agent, but we have to be very careful not to cause injury or harm to the host. So before we administer any of those antimicrobial drugs, we need to consider a couple of things. First, we need to learn as much as we can about that microorganism that's causing the infection. If we can't identify the exact causative agent, we're going to hopefully be able to figure out at least which genus it belongs to if it's a bacteria, what group or classification if it's a virus, for instance. Once we know as much as we can about what the species or the classification of virus is, then we need to look and say, what do we know about its susceptibility to the various drugs or its resistance to the various drugs, because that too will help us make a decision in which drug to prescribe. And then we would be remiss if we didn't actually think about the patient itself, because remember, we don't want to cause harm or injury to our host, so we need to look at the medical condition of the patient to determine if that's going to impact what we prescribe. In particular, do they have any allergies to any of these medications that might cause them to have a reaction? So let's start by looking at antibiotics. Now, antibiotics are naturally occurring or synthetic substances that can inhibit the growth or kill various microorganisms. Now, they play a crucial role in the treatment of bacterial infections. Antibiotics target specific bacterial structures or functions, such as inhibiting the cell wall. They may inhibit cells' protein synthesis. They may interfere with DNA replication or various metabolic pathways, such as folic acids, or even interfere with cell membrane functions or structures. So we're going to look at 6 antibiotics. Now, I know there are a lot more, but these are ones that are just pivotal for us to discuss and provide enough different examples for us to have an overview for the course. So let's start with the one that started it all itself, and that was penicillin. Now, penicillin was first discovered by Alexander Fleming in 1928, and with its discovery, we began a whole new era with medicine. So penicillin works its mode of action by interfering with the bacterial cell wall. And what it does is it inhibits the enzyme which allows those NAM sugars to cross-link. So by disrupting that peptidoglycan synthesis, the bacterial walls become fragile and they become more vulnerable to lysis. Now, penicillin was first used for treatment of syphilis, and then today it's used for all kinds of things, including streptococcal infections. It's very effective against those. So let's move to bacitracin. Now, bacitracin is also going to interfere with the cell wall synthesis, but other than that, it has almost no other similarities to penicillin, because bacitracin is one that's going to be a topical application. So it's going to be used for wounds and skin infections, but because of how toxic it is, we don't ingest it. So again, it's topical. But as stated, it does interfere with the cell wall formation. Why? Because it prevents the transport of those peptidoglycan components to be moved across the cell membrane where they're needed to be able to construct that peptidoglycan cell wall. So as we just stated with penicillin, if you disrupt the peptidoglycan cell wall, then that makes that cell more fragile, and it makes it more vulnerable to various things that can cause it to lyse. So sulfonamides. Now, sulfonamides, they're a competitor inhibitor. So what does that mean? They're going to mimic something, and in particular, they're going to mimic the nutrient para-aminobenzoic acid, which we call PABA. And by blocking or inhibiting PABA, what that does is it sabotages the ability of the bacterial cell to make folic acid, and folic acid is essential for DNA and RNA production. So you basically are shutting down the DNA and the RNA production, which means protein synthesis. Now, sulfonamides are prescribed for a variety of infections, from urinary tract infections to pneumonia. So let's look at streptomycin. So streptomycin, believe it or not, comes from a soil-dwelling bacteria, and it is going to be used mostly for gram-negative bacteria, but it blocks protein synthesis, and it's blocking the protein synthesis by binding to the 30S ribosomal subunit, and what it does is it causes that subunit to change its shape. So then the ribosome, when it comes together, is not able to read the codon. So if you're not able to read a codon, then you're going to inhibit translation. If you inhibit translation, you shut down protein synthesis, metabolic processes shut down, and then that leads to cell death. So, in one of the drugs this is one of those drugs that will often get prescribed sometimes for tuberculosis, believe it or not. Tetracycline, for instance, doxycycline is an example of a tetracycline. It also inhibits protein synthesis, but it does it different than streptomycin. So it's going to block where transfer RNA is going to dock to what's known as site A of the ribosome. So just remember that it's going to block transfer RNA from working properly. And so what that means is if you shut down that ribosome, again, we're going to inhibit protein synthesis because we're going to inhibit translation. And tetracycline is, again, going to be used to prescribe for a lot of different diseases. Everything, believe it or not, from acne to Lyme disease. Now, polymyxin is a little bit unique. It's not prescribed as often as some of the others. In fact, many times it's the last one that is prescribed. But polymyxin is, again, effective on gram-negative bacteria. It interferes with the cell membrane, in particular at the site of the lipopolysaccharide, which is LPS. So it disrupts that cell membrane at the LPS site, and LPS is obviously found in the gram-negative. So that's why it's a little bit more specific for gram-negative. So these are our 6 antibiotics we need to know for this class. Obviously there's a lot more, but do know them and their mode of action in any of those examples. So now let's shift gears for just a minute and let's talk about antiviral drugs. So antiviral drugs are going to be those chemical drugs designed to inhibit the replication and spread of viruses from the host or within the host. They target specific steps in that viral life cycle. So antiviral drugs are going to act through various mechanisms to include blocking viral entry into the host cell. They might inhibit viral replication of their genetic material, or they may interfere with the virus from leaving the cell. So again, they're going to have a specific mode of action for which they are going to work. So let's talk about 3 different antiviral drugs. The amantadine, now this is going to be one that is used for the influenza virus, and it's going to work by preventing the cell from entering into the host cell. Now, by blocking that entry into, and in particular it's blocking or preventing the M2 protein, by preventing that and preventing that virus from getting into the host cell, then what that does is that reduces the severity and the duration of flu within the host, because if the virus can't get into the cell, then it's not going to be able to replicate and divide. Because you may recall we talked about viruses, that viruses are obligate intracellular parasites. They have to get into the host cell and be able to replicate and continue their life cycle. So we're preventing them with this drug from getting into the host cell and replicating. Now, acyclovir is going to be a different type of drug, and it's used for the herpes virus. And so what it's going to do is inhibit the synthesis of the viral DNA replication. And why? Because it's going to compete with the viral DNA polymerase, and that disrupts that virus's ability to replicate its genetic material. So in its life cycle, it is preventing that DNA replication. And again, it is used for managing that herpes virus's infection. Oseltamivir is going to be used, it's also known as Tamiflu, is going to be prescribed for the flu virus. And what it's going to do is it's going to interfere with the neuraminidase inhibitor. And what that does is it prevents that virus from being able to leave that infected cell. So once that virus gets into the host cell and it begins its life cycle, this particular drug targets that neuraminidase enzyme and shuts it down, preventing the virus from spreading to new host cells or outside. So again, it's going to be significant in preventing the outbreak of the flu virus. So just 3 examples: one that prevents the virus from entering the cell, one that's preventing the replication process because a DNA replication is being shut down, and one that's preventing that virus from leaving the host cell. So this is just the conclusion for our discussion on antimicrobial drugs. If you have any questions, don't hesitate to reach out.